Hello and welcome to Lyme and the Microbiome. We're going to study how Lyme disease is affected by your gut bacteria and how your gut bacteria affect your immune system and why that might be important if you're fighting Lyme disease. So who am I? My name's Dr. Richard Matthews. I'm a chiropractic physician in practice since 1990. And I'm a board-certified chiropractic neurologist. I wrote a book about gut bacteria, athlete, father, husband, do a few things, and I consider myself a Lyme Literate doctor. If you're not sure what Lyme Literate means, it means somebody who understands that there is more to Lyme disease than what the CDC or the IDSA has to say, and recognizes the reality of chronic Lyme disease. So if you've been looking for somebody that actually believes in chronic Lyme disease, I'm your guy. Disclaimers, common sense. This is a webinar on YouTube. This should not be considered a replacement for your physician's advice. I believe the best patient is a well-educated patient, and a successful doctor-patient relationship depends on two-way communication. And just read the rest of this. Basically, by watching this, you're consenting to not make crazy decisions just because I said something. This is all about learning. This is not medical advice to replace what your doctor says. All of the statements that I make in this are based on research studies, and at different points in this slideshow, I may click on a link like this, and you can see the bibliography of research studies that I've put together that are the basis for everything in this presentation. So, what is my framework for how to improve with Lyme disease? It's based on 3 different things, and in different patients, these may be weighted with some more important than others, but the 3 pillars of this approach are working to improve your gut microbiome, that is your gut bacteria, those trillions of bacteria that live in the gut that help run your immune system, manage your brain, and do a whole lot of other things. We do that by testing, we'll get into that later, and then targeted interventions to try and improve those gut bacteria. Then bactericidal herbals or antibiotics in some cases. Now, my threshold for sending somebody to a medical physician to get antibiotics for Lyme disease is generally when a person is so bad that they're having seizures, not able to work, not able to sleep, and is at a point of literally feeling suicidal or close to it anyway. The idea being that these natural treatments, while they're really useful, they're not for everybody. It depends on what point of care you're at, and if you're suffering life-threatening effects. If you have Lyme carditis, for example, you really should start out with antibiotics, and after you can't get those, then you can transition to this type of approach. So this is not meant as a treat everybody this way approach, just for clarity. Lyme and co-infection testing. There's a whole lot we can talk about there. I'm just going to start out at this point by saying that the most commonly used Lyme tests are not nearly up to date, not sensitive enough, not interpreted correctly, and there are much better options, which you will see shortly. How widespread is Lyme? Even this is controversial. This is the CDC's map leading up to 2019. You'll see in some areas like Arkansas, I did practice in Arkansas for a number of years, and it's in the lowest category, 29 to 51. Notice this is reported cases. Reported cases means cases that are reported to the CDC. That requires 2 things. It requires a doctor to actually diagnose Lyme, and it requires the CDC to accept a report. Now, when I practiced in Arkansas, and I tried to submit positively diagnosed Lyme cases to the CDC, they didn't accept cases from Arkansas because they didn't believe there were any, so they wouldn't accept any submissions. So guess what? At that point, this was 0 for Arkansas, which is a little bit like when there's something you're looking for in a store and it's not on the shelf, and the store manager says, well, it's not on the shelf because we never sell any of it. And you're like, well, duh, of course you don't sell any of it. It's not there. I can't buy it, or you would have sold one. So if the CDC will not accept submissions, guess what? The numbers are low because this is total of submitted cases. Most of these are normally reported by medical physicians, and if there are very few medical physicians that believe Lyme disease exists in the state, then you're going to see very low numbers. So this map may be completely fictional. When I moved to Maine and practiced for 6 years in Maine, I saw a huge number of Lyme cases, but also was still in contact with some of my patients that I'd practiced on, that were in my practice in Arkansas. And for example, one of them told me she flew to California, got horribly sick on the way, and in California ended up in the ER, where a doctor from Maine was attending in the hospital in California. He ran a lab test in California and diagnosed her with Lyme. She went back to Arkansas with her positive Lyme test from the MD at the hospital. And her doctor looked her in the face, she said, and told her, that's not possible. There is no such thing as Lyme disease in Arkansas. So there you go. I could go on and on about that. Ixodes scapularis is the tick that normally carries Lyme disease, carries Borrelia burgdorferi, the bacteria that normally causes Lyme disease, although there are several others. Most people call them deer ticks or black-leg ticks or a number of other things that we won't repeat here. But if the tick that bit you is a dog tick, these can still transmit Lyme and co-infections. It's important to note that because a whole lot of doctors will tell you that that's not possible, but I'm here to tell you it is possible. I've seen it. Causes of confusion. There are many. I'm just going to try to highlight these. If you've been there and seen it, you may be cheering like, yeah, I've seen that, or this may be completely new to you. I'm going to try to condense a huge subject into one slide, so forgive me if I'm brief, or try to be. If you've been told that your Lyme test is negative, you may still have an infection. How's that possible? Well, different doctors have different points of view, different doctors follow different guidelines, and different doctors use different labs and different lab tests from different labs. And some of those are more sensitive than others. The CDC, IDSA guidelines say to only use the 2-tiered test, which is an ELISA antibody test, and then only if that's positive, a Western blot. There's a high threshold for a positive test. That means you have to have, they call them bars, you'll see this in another slide later. You have to have at least 5 of them for a positive test. ILADS considers it positive if there are only 2. And the test that I use provides a whole lot of additional information besides just the Western blot. So ILADS, International Lyme Associated Disease Society, also supports the idea of a symptom-based diagnosis, meaning if you're sick enough and you had a tick bite, you probably have Lyme. They downplay the importance of lab tests, which is good when talking about this 2-tier test because it's, in my opinion, not a very good test. But they don't really make acknowledgement of more modern tests that are done with better protocols, better equipment. They do recommend antibiotics for months or even years sometimes, while the CDC IDSA guidelines say that if you get antibiotics at all, it may be as little as a day or 2 weeks, and rarely more than that. The approach that I like to use is based on reading research studies and using what's appropriate from these other protocols. I like reliable, validated testing that has been well researched, that has published research, and that's available. The 2-tier testing provides a lot of false negatives, which means if it says you have Lyme, you definitely have Lyme. If it says you don't have Lyme, there's still a significant chance that you do have Lyme. There are also studies showing that Borrelia, the bacteria that causes Lyme, cannot be completely killed with antibiotics. So the idea of prescribing antibiotics for weeks, months, and years is a little bit flawed when you don't also focus on doing things to build up your immune system and your health, your strength. You can also get a negative diagnosis and still have Lyme disease if they're testing for only one species. There are many species, 17, I think, on the tests that I use. And you might just be sick with a different species of infection. You could have not Lyme disease, but a Lyme co-infection, or you could just have a really, really imbalanced microbiome, often as a result of taking antibiotics for months or years. Why is the microbiome? What is it and why is it important? Well, trillions of gut bacteria live in your gut. They are necessary for the development and function of brain, nervous system, immune system, your HPA axis, hypothalamus, pituitary, adrenal. HPA determines the sensitivity of your body's fight or flight response. So if you have a lot of anxiety, if you startle easily, if you wake up in the middle of the night amped up with your heart going a mile a minute, it's a good possibility that your HPA axis is stuck in fight or flight mode and can't find its way back to relax and digest mode. Your microbiome influences brain function, emotions, autonomic control. I wrote a book about the gut bacteria. Here it is on Amazon. It's called The Symbiont Factor. You can also pause this video and just do this QR code, scan the QR code. Here's a research study that's one of the better known ones about controlling inflammation. Bengmark was a liver surgeon who found that after 30 years of studying this, that if he didn't kill off a patient's gut bacteria before doing liver surgery, they lived a lot more of the time. Oops. So many of the immune responses that you need to kill Borrelia bacteria depend on your microbiome. There's something called neutrophil extracellular traps. Now, if that sounds really sciencey and wordy, let me warn you, there is science involved in this presentation. If your brain glosses over, you can just fast forward. I'm going to put an index down in the description so you can skip to whichever section you need and use this video as a resource, because this is going to be a long video. So buckle up. Here we go. Neutrophil extracellular traps work a bit like a fishing net, and they're made from neutrophil DNA. Those are one of your good guy immune cells that help kill things. Here's a research study about it, showing neutrophil extracellular traps entrap and kill Borrelia burgdorferi. And they're not affected by tick saliva. But those neutrophils require aging. It's like a good cheese or a good wine. The microbiome is part of what helps these neutrophils to actually achieve competence, and this is worth sharing. This is what I do. I read research studies and learn things and use that to help patients. So I'm going to show you a lot of these studies as we work through here. There are a lot of other immune factors necessary, like invariant natural killer T cells, interleukin 8. One of the things that's key here is understanding that Borrelia bacteria can completely avoid your immune system's response, and they do that by causing inflammation. It's a little bit weird, but this bacteria can use your immune system against itself, against your immune system, to escape. So what is the Lyme life cycle? This bacteria is interesting because it goes through a number of different hosts. And then at one point there are larvae in the ground. Those become nymphs. They infect a variety of animals. They become adults. They attach and feed on the third host and then lay eggs, and the eggs hatch and become larvae. So you can see there are a number of animals, and humans can end up in here also, where the tick feeds on us. But normally, mice, birds, squirrels, rodents, and deer are the biggest ones. And the researchers really emphasize the deer and mice are the most important hosts to this life cycle. Now, what can you do to interrupt the life cycle? One of the things to realize is that birds carry ticks. If you're looking at this on your phone, this is a picture of a bird's head with a whole bunch of little ticks around it. It's really gross. Because birds carry ticks and birds fly pretty significant distances, one of the things you might consider is do not feed the birds and attract birds to your house because they will bring ticks to your house. And when you look at that life cycle, one of the things you're seeing is the bacteria gets in the local biology. So if you import a whole bunch of ticks to your yard, your property, then you're inviting that bacteria. In addition to that, cats, for thousands of years, cats have been helping humans to get rid of the rodents that live with us. They live with us probably because of that. Some of them kind of like us, but they're cats. They're pretty independent. You should get a cat because cats are amazing at killing mice, and that interrupts the food chain, the biological life cycle of Lyme disease. In addition to that, deer carry ticks and deer carry Lyme disease. Do not feed the deer in your yard. Do not feed the deer in a pasture. Let the deer be. Let nature have their way with the deer. If you feed deer through the winter, for example, the does will end up having twins. They have twice as many fawns, and that can quickly lead to overpopulation. Where I practiced in Maine, if you look at a map of Lyme disease in Maine, it's primarily along the coast, and that's primarily where people feed the deer. And it's primarily where if you're driving around, you see way too many deer. Like they get hit by cars all the time because there's an overpopulation of deer. Mostly because people feed them, and also because many times those same people prohibit deer hunting. So they have twins and nothing eats them. So you have a whole lot of deer. Deer carry ticks and Lyme disease. So you end up with a whole lot of Lyme disease. Don't feed the deer. Don't feed the birds. Get a cat. Interrupt that life cycle so you have less Lyme in your life. What if Lyme disease could be transmitted by mosquitoes? Well, that would be terrible. There is actually some evidence that it can be. I think part of this comes down also to how vulnerable you are to Lyme disease. And there is a significant difference in vulnerability between individuals, and probably how many mosquito bites you get and how long they're on you. If you think about it, a mosquito is not biting you for very long compared to a tick. A tick can stay there 2 days. A mosquito, maybe 30 seconds. But despite that, mosquitoes do use saliva with an anticoagulant and an immune complement inhibitor, just like ticks do. So there are a number of researchers that published a study indicating, yeah, it's not impossible. But even worse is, what if Lyme is sexually transmissible? I've actually inferred an example of this in my practice, where one patient that I was treating for Lyme disease apparently transferred it to her boyfriend, and he got Lyme disease even though he did not have any history of tick bites whatsoever. If you look at Lyme, it's a corkscrew-shaped bacteria. It's called a spirochete because of that shape. And it gets into us normally by a tick bite. But if you looked at another spirochete like Treponema pallidum, which causes syphilis, they look really, really similar. And they're both optimized for getting through mucous membranes. And if you look at the research on it, Borrelia spirochetes have been identified in female and male secretions. So it is technically possible. In the Lyme community, on Lyme forums, there are a number of people who have said that it's actually happened to them. What happens during a tick bite? The tick bites you and kind of numbs you and thins your blood with its saliva, so you usually don't notice it. and it can suck your blood because the blood won't clot. That tick saliva inhibits your immune system. I mean, and then after the tick saliva inhibits the immune system, the Lyme bacteria will actually use that tick saliva to help evade your immune system. That's the first step of invasion. Now, this is a chart that I made up to chart how Borrelia bacteria move through the body. This is a whole other presentation, but I'm just going to give you the punchline slide here. Skin, blood, joints, brain, and moving to the right, that axis is time. So there's the infected tick. It bites you. The salivary protein of the tick helps the Borrelia bacteria get into your blood. That stimulates interleukin 10, which inhibits inflammation. It's part of why you don't feel the tick bite when it's in your skin. The tick produces an outer surface protein C, that prevents your macrophages, your white blood cells, from eating it. Then it gets into the blood. This is usually where you feel sick with what people describe as a flu-like illness. Hurt all over, get a fever, generalized achiness. Feels a lot like a recent viral infection. There's a lot of overlap in symptoms there, except for the respiratory ones. The tick will the bacteria will actually trigger inflammation to occur. It'll trigger your immune system to create inflammatory gamma interferon. And the tick, the bacteria uses that to activate a gene sequence so it can change its outer protein. And that means your body has to start all over producing antibodies. Then it makes your immune system produce inflammatory cytokines, interleukin 23 and 17. Those make your joint capsule and your blood-brain barrier more permeable so that the Borrelia bacteria can get through those capsules. Effectively, what it's doing is it's trying to hide from your bloodstream because that's where most of your immune system is. But once it gets into your joints, you're going to have joint pain. And once it gets into your brain, you're going to have, neuroborreliosis, which can cause anything from brain fog to loss of muscle control, seizures, mood changes, psychiatric problems, almost anything brain-based, neuropathy. And when you look at testing, if you're trying to find it in the blood with conventional testing, this is a weakness because you're only going to succeed at doing that if the test is done after it gets to your blood and before it's left your blood. When you look for other antibodies and use PCR, other technologies like more advanced tests use, this is not as much of an issue. Bullseye rash. Some people get a bullseye rash. This is a rather stunning example of bullseye rashes. Some people get hardly any rash at all after a tick bite. Many physicians mistakenly believe that if there was no bullseye rash, there's no Lyme infection. Not true. Some of these studies show that as few as 33% actually ever get a rash. Many masks of cutaneous Lyme disease. Anyway, that says it often looks like something else, or the rash may look different. Is everyone susceptible? Not really the same way. Some people have a good enough initial immune reaction that it reduces the chance of long-term infection. A lot of the immune factors that you need to fight Lyme depend on your gut bacteria, your microbiome. Some people just don't get any symptoms at all, and some people get sick rather quickly. There's a big difference in genetic susceptibility in some people, and there are attempts being made to figure out what is causing that difference. And some Lyme-resistant mice in research have higher levels of interleukin 10. Interleukin 10 is something that reduces inflammation. And in this study, if they blocked interleukin 10, the mice became more likely to get Lyme disease. And guess what? your microbiome promotes interleukin 10. That's in response to a different infection, but it's still what it does. Now let's look at testing for a moment. Most tests, the CDC IDSA protocols that the majority of physicians use, looks only for one species, Borrelia burgdorferi. Actually, it's often one strain of it. No co-infection tests are done typically. And in Maine, for example, 48% of the ticks carry co-infections. They're only looking for one antibody with the initial ELISA test. So if you don't have that one, the testing ends and you're told that you're negative. This misses a lot of cases. I've seen patient reports from many patients that their physician would not even do a Lyme test because they did not have a bullseye rash, or they would not do a Lyme test because they had already been treated for Lyme in the past. The test that I use is Vibrant Wellness Tick-Borne 2.0. That panel looks for 17 different species of Borrelia instead of 14 different co-infections, 9 different opportunistic infections, 2 different types of antibodies, short-term and long-term, IgM and IgG. Also uses PCR or polymerase chain reaction, which is a different test technology, to look for Borrelia and co-infections. It's much higher sensitivity and specificity, and you can also use it to test for reinfections. There are a lot of limitations to ELISA testing. It's thought to miss as much as 60% of acute cases. It's really an outdated test compared to more modern techniques. Western blot is pretty easy to fool because different proteins have similar molecular weights, so they can overlap and just make it a less reliable test. Immunoblot has the same limitations as Western blot, and traditional PCR is low because, again, you're only going to see a positive PCR if you catch it while it's actually in the blood, because with PCR, you're looking for the organism itself. It's good to do the test, and it's part of the Tick-Borne 2.0, but it's not the only thing that the whole test result depends on. Tick-Borne 2.0 has IgM and multiplex PCR, which is done on a microchip, that they can amp that 16,000 times, which makes it extremely sensitive. And it also includes testing for co-infections. This is perhaps one of the most common reasons that people are sick and their doctors say, no, you must be fine. It must be in your head. It couldn't be tick-borne because your Lyme test is negative. Well, if you didn't test for co-infections, what if it's not Lyme, but it's a co-infection? Some of those can make people just deathly ill. Anyway, Vibrance tests are done on a microchip, an immunochip, which has been scientifically tested and research validated. It has an extremely high specificity and sensitivity. The two different criteria here are that they give you an answer based both on CDC and alternate, like ILAD's criteria. So you get both perspectives with this test. But 100% sensitivity, 95 to 97% specificity is crazy high. It's an extremely reliable test. So here's a saying: if you're not testing, you're guessing. In my opinion, anybody who has Lyme symptoms should take this test and see what it is that they are fighting. It's really, really important. Here is a quick peek at all of the different organisms that are tested for in a Lyme, in the Tick-Borne 2.0 test. If you want to read all of them, just freeze the video and read them. There are a lot of them. The amount of information in the report and the interpretation matters also. I'm going to give you a quick peek at a sample report, which I'll start at the top here and just kind of fast scroll through it. It starts out with a summary, things that you tested positive for. And then it goes through a color-coded chart that shows you the level of antibodies for each category and each organism. And it's a very, very thorough test, as you can see. And it's backed with a large number of research studies to validate it, 77 of them. It's a great test. So what happens after an infection? Borrelia burgdorferi, like everything else, is just trying to survive and reproduce. But don't feel sorry for it. That's not what I mean. Just everything tries to survive. Your immune system responds, creates inflammation, tries to kill it, and does not succeed in a lot of cases because the bacteria is able to avoid your immune response by making it produce gamma interferon. Now, interleukin 10 is one of the things that controls inflammation so this doesn't happen. And your invariant natural killer T cells, ink T cells, keep Borrelia from going into your joints and brain, really so that your immune system can keep killing it. And this is one of the most critical research studies here. And you can see the full study by clicking on this free link. This is available to the public. It's not some special login. And these killer T cells act as a barrier. Now, extravascular means they keep the bacteria, they keep the Lyme bacteria in the blood vessel. Now, that's important because that's where your immune system does most of its work. And this part of the immune system corrals all those bacteria so the rest of the immune system can kill them. How effective are preventive antibiotics after a tick bite? If it's right away, research shows it can prevent 74% of infections. 24 hours after the bite, that's a little less than half. After 48 hours, does not work at all as prevention. So if you're looking at a 48-hour time window, you're no longer preventing an infection, you're treating an infection. Now, the problem with antibiotics is that they increase gut dysbiosis, they imbalance your gut bacteria, and they increase the risk of antibiotic-resistant genes developing, which is bad because then when you need antibiotics, they aren't going to work. The biggest issue also is that Borrelia bacteria are very, very resistant to antibiotic, and they form persister cells, cysts, and biofilms, plaques in other words. And those basically mean these drug-tolerant persister cells. Really what that means is that your antibiotics are not going to kill 100% of these bacteria in many, many cases. And, you know, it's not just one stray researcher that found this. It's really, really several. And there are just 4 that I picked out here. This one has the most complete explanation. That's Bernson. What that really implies is that if you're relying just on antibiotics, you're probably going to end up with a chronic Lyme infection. If you take antibiotics for weeks and months, you're going to end up with a really, really imbalanced gut bacteria. That's important because you need your gut bacteria in order to prevent infections. So when you stop the antibiotics, what prevents the infection from coming back? Since we just said the antibiotic doesn't kill all the bacteria, when you discontinue antibiotics in 6 to 12 weeks, those bacteria can revert to an active form. If they don't, it's because your immune system is working well, so they're not going to be able to. If you've destroyed your gut bacteria, your immune system goes with it. So this is what a Western blot looks like. I just put it in here as an example. And this goes through a deeper explanation of how outer surface protein C is one of the ways that the bacteria avoids your immune system. Now we're going to go through an even more detailed example of how the bacteria avoids the immune system and why the microbiome is important, and how antibiotics, while you may need them in some cases, can also make some parts of this much worse. If you don't want this much science, feel free to skip ahead. But if you're fighting Lyme disease, I really believe you should know your enemy. So, red arrows means that something is inhibited, and I've tried to make the new things on each slide yellow. So, all of these antibodies help kill Borrelia in your bloodstream. These are some facts about your antibody response. While gamma interferon does initially kill some Borrelia, it also allows the bacteria to recode its outer protein expression so the antibodies don't work anymore. Bacterial colonies can communicate between all the members. There was a researcher named Professor Eshel Ben Jacob who really made a convincing case that a bacterial colony acts with the intelligence of a multicellular organism. Even though each individual bacteria isn't much, they communicate using something called quorum sensing. And all of the bacteria in the body that are the same kind of bacteria can all act as one, which is a little spooky. Invariant natural killer T cells stimulate all of these beneficial antibodies. And if you're watching the slide presentation, you can actually click on all of these and see validation and read the complete full study if you want to take the time to do that. I'm just showing you it's there. It's not just me saying this. Your gut bacteria, your gut microbiome produces something called alpha-galactosylceramide, which is actually a little bit inflammatory, but that helps stimulate your invariant natural killer T cells. It's something called a glycosphingolipid. Yeah, that's used in the outer membrane of the symbiont bacteria. And some of these bacteria, then this is one of the mechanisms where they stimulate the immune system to produce more antibodies that you need. And those antibodies are essential for the developing immunity against this infection. They also stimulate some other immune factors because you have nerve endings that go down into the gut, into your intestines, that talk to your gut bacteria. There's a communication back and forth with the nerve endings. And that's part of what induces these immune factors. Your gut microbiome also boosts an immune factor called CD8. CD8 also stimulates your invariant natural killer T cells. So you can see that there's a chain reaction of events here happening. Now, Borrelia makes a defensive move here to try to get away, and it triggers the release of interleukin 23 and interleukin 17. It's making your immune system open the door for it to escape the bloodstream, which is a little bit freaky that it knows to do that, but there you go. And then it is able to escape. Now, let's kill it with antibiotics. Sounds like a good idea. Borrelia must die. What could possibly go wrong? You already know the punchline to this. The antibiotic also reduces your gut bacteria. Reducing all of the goodies that we just saw were resulting from the gut bacteria. I have seen patients, two in particular, hello, if you know who you are, who had been on years of antibiotic therapy. And when I evaluated their microbiome, their normal gut bacteria were less than 1% of normal. And over 90% of the bacteria in the gut were one species of pathogen, which is really beyond super imbalanced. And these were people that every time they got off antibiotics, they had Lyme flares again. Antibiotics deplete gut bacteria really, really effectively, and that is a bad thing. It can deplete them enough that they do not come back ever, which is worth considering. Interleukin 17, that makes your blood-brain barrier and your joint capsules leaky, and that's how the bacteria gets into that joint space and brain space. Lots of researchers have shown this and demonstrated what it does. Here you go. Some of these studies are just showing the effect of interleukin 17 and interleukin 23 on those barriers. And some of them specific to Lyme. Interleukin 10 is the good guy. Your immune system produces that to reduce inflammation. It inhibits this inflammatory interleukin 17, so it helps protect your blood-brain barrier and joint capsule so you don't get Lyme brain and your joints don't hurt. But what is it that boosts interleukin 10? We want it because it inhibits Borrelia burgdorferi, IL-17, and Lyme arthritis. Your gut microbiome stimulates interleukin 10. So if you look at this diagram, the bottom here connects back to the top. If this were on paper, you could roll it up in a tube. So interleukin 10 inhibits the inflammation, and it keeps the bacteria from hiding, so the antibiotics or your immune system can kill it. This is really, really useful if you're trying to get over Lyme disease. But what happens if your microbiome is depleted from the antibiotic Lyme treatment? Long-term recovery requires a diverse microbiome, and you lose that. Your cystic form of Borrelia and secondary infections are just waiting for your immune system to be down in order to come back. And that happens really easily if your antibiotics wiped out your gut bacteria. So this is a big reason for rebuilding the microbiome. You can do some of this during antibiotic therapy. You definitely need to do it after antibiotics. Or if you're going the natural route and not using antibiotics, work to improve your gut bacteria, which, yes, that means you probably need to look and see how is your gut bacteria before you start to improve it. Now, there are some substances that can help. Huperzine is a nutritional, it's a food supplement that helps to strengthen the blood-brain barrier. It's good stuff. We also use it for concussion patients. It reduces cognitive deficits. Let's see, what's this other one say? Huperzine is neurovascular protective. Research studies can actually be interesting. And Huperzine offers some protection similar to glucocorticoids drugs. Curcumin is another thing. It inhibits inflammation, inhibits both interleukin 23 and 17. It helps protect your blood-brain barrier. Let's see. I think this is one of the studies to show. Yeah, there you go. Helps CD4. And come on. There you go. So curcumin is good stuff. There are a lot of different brands of curcumin. It is not the same as turmeric. That's kind of a peeve of mine. People say, oh yeah, I've been taking turmeric. Well, turmeric only has 4% curcumin, and curcumin itself is 95% curcuminoids, so it's 19 times as strong as turmeric. Curcumin also helps boost interleukin 10. It protects the joint capsule and blood-brain barrier, which is very useful. So here's the mostly complete picture. Kind of crazy, I know, but you can go back through it, slow motion it, speed it up, whatever you want, so that you can see the chain of events here that shows that your gut bacteria are very important for fighting Lyme disease, for these specific immune factors, and that curcumin and huperzine are very useful potentially as well. Now let's go back to the subject of Borrelia not being killed by antibiotics. What is a biofilm? It's a plaque. Now, one of the researchers that's done the most about this is Alan McDonald. And he also has done research on these spirochetes in degenerative disorders like Parkinson's and Alzheimer's. So you should really go through these studies and at least read the headlines and understand what they are, so that you know what the research science world has actually proven. Because you may hear all kinds of things, but what do we know for sure is really quite important. So what allows Lyme disease to return to activity is really when your microbiome is depleted, when your immune system is depleted. Stress can also do that. Sometimes it just reactivates when somebody gets older. Now, if you consider that, what are the biggest uncomfortable parts of getting old enough that a lot of people experience as symptoms? Brain fog, memory problems, trouble with movement, joint pain. So really, if you consider it, how many of those symptoms are actually mandatory to come with the aging process? Maybe not as many as we thought. Here's a picture from Alan McDonald's work of one of these cysts and plaques forming. It's from the movie Under Our Skin, which if you really want to understand how Lyme affects people and how doctors are falling short in trying to help, this is a great movie to watch. Follow the link, watch the movie. Lyme is a great imitator. Alzheimer's has been found to be caused in some people by infection, particularly with Borrelia. Parkinson's also. MS also. These are important because most people with these diseases never receive a Lyme test, never receive an autoimmune antibody test like a neural zoomer test. How many people with brain inflammation issues might actually have Lyme disease? How many depressed people or people that just think that getting old is horrible might actually have Lyme disease? We don't know because we're not testing enough of them. Here again, curcumin, huperzine, very useful. Antibiotics damage the gut bacteria. There's some studies going further into that. Now again, this doesn't mean never take antibiotics, but not everybody with Lyme needs antibiotics. Some people definitely do. Compared to the antibiotics, doxycycline and cefuroxime, there are a couple of herbs that were found to kill Borrelia more effectively in research, so that's worth considering. Japanese knotweed, wormwood, cat's claw, Ghanaian quinine, which most of us in the Lyme community know as cryptolepis, but that's really what it is. Ghanaian quinine is used as an herbal remedy for malaria. And black walnut, which was an old-time remedy for worms. Here's the research study. And you can scroll down to the abstract, and I just have to show you this. Cryptolepis and Polygonum cuspidatum showed strong activity, and in subculture studies, 1% solution of Cryptolepis caused complete eradication when doxycycline and cefuroxime could not kill the Lyme. So I would consider that. Yeah. When you have published research showing it actually kills the bacteria better than the antibiotics that a lot of people fight to get. Now, what do you want to do? Here's testing. Tick-borne 2.0 is the blood test I showed you earlier that helps you detect Lyme. I like to say if you don't know what you're trying to kill, you won't know what products to use to kill it. Because when I find an infection, the first thing I do is to research what kills this. Gut bacteria testing is also super, super critical. The test that I use is called GutZoomer 3.0. Here's a quick peek at it. This test, just like the Lyme test, goes through a tremendous amount of detail, 171 different organisms, shows you different levels of them and how it affects your disease risk, what's elevated, what's depressed, what probiotics you might need, what prebiotics you might need. And it also goes through, toward the end here, sorry if I'm making you dizzy, it goes through parasites. Do you have worms? And single-celled parasites, protozoans. And gut pathogens. Do you have any pathogenic bacteria? Antibiotic-resistant genes, fungi, viruses, candida. It's all in this test. It's an amazing, amazing test. Typically, when one of my patients does this, we do a Zoom review afterward, and I screen share the lab results. They'll already have the lab results in front of them, and I go through all the results and explain what it means and what to do about it. Takes about an hour to go through one of these on a Zoom call. So, in summary, Lyme is caused by Borrelia bacteria. And there are a lot of different species of Borrelia. Borrelia needs inflammation to succeed at infection, and it uses that information to evade your antibodies. Reducing, controlling inflammation is part of the process that I recommend to help your antibodies, your body's immune system, to try and kill the infection. Improving gut bacteria helps that. And there are some herbs that can be very, very useful. 2 tests that I recommend for anybody with Lyme disease. What can you do next? Well, I have 3 resources here for you that you should pause this and you can visit the links, or if you're watching on a computer or tablet or YouTube on a TV, you can freeze this and just use your phone and read that QR code and it'll bring you right to the website I'm about to show you. This is my online store. This is just the sign-in screen I'm going to show you here, but it's really simple. You just create an account so that you can place an order. You can order your own Lyme test directly from this website, unless you're in New York State. My other online store has a whole host of supplements. Again, you just create a free account here. There's a registration code right here, RM1472, that you can use. That you can use to create your account. And here's my actual website, which has a scheduler where you can choose what kind of visits you want to schedule, and you can pick what day you want to schedule it and actually see what times are available and plug yourself in. I practice virtually online over Zoom, do a lot of email also, and communicating through the portal, the lab portal. And most of my patients are not local to me. And I can do that for anybody except people in New York State, because New York State requires practitioners to have a New York State license, whereas telemed is easier in most other states. This website goes through a few explanations about what I do, and it provides more information about how to get tests ordered. So there you go. If you made it this far, God bless you. Even if you had to do it in bits and pieces. There's a lot of information here. I hope you found it useful. And I wish you the best on your Lyme healing journey. Please contact me if you would like some help, some guidance. If you want to just book a session to review what you're doing and see if you're missing anything. If you're interested in doing this lab work but have questions, schedule a time. I'm there to help. Thank you.